Each sample received at NAC consists of about 200 tubers. A sample is registered digitally in advance, with the grower specifying what the sample is to be tested for. When the samples are received, they are scanned and numbered. Then, in the laboratory, the potatoes are deposited on a belt. Here, the sample is scanned one more time and also labelled with various stickers for follow-up processes. This is important for the traceability of the sample and the results. The entire procedure is carried out under this number. In the cutting room, 2 pieces are cut from each potato, a peel and a heel end. These are placed in separate cups. The result is 2 series processed separately in the laboratory. If the sample is tested for brown rot or ring rot, buffer is added to the sample. The samples are placed on a shaker overnight. The samples are then centrifuged and a fraction of the material is transferred into a tube. From the tubes, the material is transferred onto a specimen slide. The specimen is stained so that the slides can be viewed under the microscope. This is a key step, as the staining enables detection of brown rot or ring rot. A special light, known as immunofluorescence, reveals whether there is brown rot or ring rot in the sample. In suspected cases, the sample is tested by the NPPO, the National Plant Protection Organization, for confirmation. When the sample is tested for viruses, water is added to the samples in the grinding lab. The samples are then put into large shakers, where the contents are milled into juice. This juice is transferred into tubes. A full block of tubes goes to isolation. Isolation is a technique in which the genetic material of the virus you want to demonstrate is extracted from the juice. This is needed for a PCR test. Using a pipetting robot, the materials needed for isolation are transferred. Then a machine, the Kingfisher, helps to isolate the genetic material from the juice. A PCR test is then carried out using this isolated material. This is the step that shows whether there is a virus in the sample, and if so, which one. The result is assessed separately by 2 different people. If the assessments match, the result is released.